Hello students, welcome back to my YouTube channel LBioTutor. Today I am going to talk about DNA replication. This is very important topic in A-level biology. So let's begin it. So what is DNA replication? DNA replication is the process of copying of double-stranded DNA. Here we will have two identical copies of DNA molecule. So mother strand, we will have mother strand as we are having it, and the double-stranded mother strand will become two, that separated the two DNA molecule, and the mother strand will give two separate double-stranded DNA molecule as this picture. And the process of DNA replication is similar in prokaryote and eukaryote, but the involving enzymes are different in structure. In this figure, you can see the mother DNA molecule and how it become replicated in as result in two identical DNA molecule. So what are the importance of DNA replication? Or otherwise you can say copying of DNA. So first thing, information for the life is in DNA. As we know, inside the nucleus we have the information for the process of all the process of the information is inside the nucleus. So these new cells receive this information from parent cells. That is more important. Also in multicellular organism grows by addition of new cells. Cell number is increasing. For this purpose, DNA replication is important. If you have any damaged cell or dead cells, this will replace by new cells. For the formation of new cells, DNA replication is important. In asexual reproduction, offspring is identical to parent cell, and the DNA replication will result in genetic information going to the daughter cells. For this situation, mitosis is held at the process of cell division. I have discussed about mitosis in my previous video. You can watch the video and see what the mitosis. In sexual reproduction, it is resolved with meiosis. Because of the meiosis, the number of chromosomes is constant. From the mother generation to this parent generation to daughter generation, the number of chromosomes is constant due to DNA replication. Also, there may be some errors in DNA replication. We call it mutation. With this mutation, there may be different features in the species. So some organism may have different species rather than other organism in the same species. This will result in variation, different characters. This is resulted in evolution of the organism, and the ultimate results of the replication is to maintain of the life and continuation of the species. That is the ultimate importance of DNA replication. If we talk about the DNA replication process, it is very carefully controlled and it is coordinated by enzymes. Dear students, this is very important to understand what are these enzymes and the process of the replication. DNA synthesis. For the synthesis of DNA, parent DNA strand is used as the template. Parent DNA strand is used as the template. New DNA double helix contain one parent DNA strand, and the other one, the parallel one, which is antiparallel to the parent DNA. So that is new complementary strand. So you can understand it now. So you have double strand DNA. We suppose that it is uh, 1 and 2. The strand 1 will prepare a complementary strand as 1 dash, or strand 2 will prepare a complementary as 2 dash. Ultimately you will have two identical DNA strands. We know that DNA is tightly packed in DNA. DNA tightly packed in cell as chromosome. I have discussed that thing in my video about DNA, so you can watch the video. So because of that tightly bound situation of the DNA, we have to relax that. First thing, DNA should be relaxed. This double helix DNA should be separated for the replication process to start, and the helix will be separated at a specific sequence of DNA. We call it a origin of replication. What is it? Origin of replication, or otherwise we call it as a ori. So DNA replication start at origin of replication. So entire circular DNA replicated in both direction. So it is happening in the both direction, the prokaryotes especially. Okay. So new DNA strand can move in only one direction. This is 5 prime to 3 prime end. So if you take as this figure, first time 5 prime end is at the, when you're talking about the upper DNA strand part, 5 prime end at the left side and the 3 prime end at the right side. So the synthesis of new DNA starting at the where you have the 5 end, 5 prime end. If you have 5 prime end, then ultimately it is there at the 3. So you will starting with DNA, the nucleotide you put into the 5 end. You put in the 5 as 5. 5, 5, 5. It's going to 5 to 3, starting at the 5. So this will continue in one side. If you go into the other side, what happened in the other side of the strand? It is as the 3 to 5. That side you have to go from 3 to 5. This thing won't happen. So you have to put the nucleotide into 3 end. To the 3 end you put the 5 end of the nucleotide. So in the other side it will become lagging strand. So 5 to 3, 5 to 3, that's small uh, part by part it will replicate. We will see the procedure later. So one new strand can be synthesized continuously. You call it leading strand. And the other side of these fragments, small fragments is replicating. You call it a lagging strand. So these fragments, we call them Okazaki fragments, which is in the lagging strand. So basically there are 5 enzymes and one specific protein which is important in DNA replication. So what are the enzymes? Helicase, topoisomerase, primase, DNA polymerase, DNA ligase, and the protein is the single strand binding protein. So with the using of these enzymes, let's see how this process is going. So while we are learning the enzyme, we can understand the process. We will see it. Helicase. Helicase. We know that double-stranded DNA is alpha helix structure is there. So we have to unwind the double helix. So unwind double helix is done by helicase, and helicase also help to separate the two strands. For this process, we have to break the hydrogen bonds, and this is the process of using ATP. There you have to use the ATP as the energy source. So with the using of helicase, we will have two strands of DNA. This will using as the template for the DNA synthesis. The second one is student. The second one is topoisomerase. This is working ahead of direction of DNA synthesis. This is also very important because this will introduce break on one or both strand of the DNA. We know if there's something with twisting, if we try to untwist it, there's more chance of twisting it more in the other side. To prevent that one, we have to cut that, or otherwise you have to lose the other side also. So this is done by topoisomerase. It is done by temporarily cutting the strands and attaching them in again. So twist that molecule to reveal the strand, then release the curtain. That is done by topoisomerase. So we have to talk about helicase, topoisomerase. Third enzyme is primase. Actually, it's a RNA polymerase. Primase is producing a polymer. RNA is a it will produce a primer, something called primer. Actually, this is a RNA fragment. It's a small RNA fragment which is having 8 to 9 RNA. But very important, student, this is RNA, not the DNA. So it is RNA fragment. So this primer attached to the place where the DNA replication is starting. You call it origin. The primer is going to the sequence which is there in the DNA strand. With the using of primer, we can start the DNA polymerase action. We will discuss it later. So the primer will facilitate the action of DNA polymerase. And now the DNA strand is producing with nucleotide. So this nucleotide added to the 3 end of formed nucleic acid chain. Right. The primase, what is the function of primase? It is a RNA polymerase which is producing primer. It is a RNA fragment. DNA ligase, that is the enzyme which is joining new synthesis adjacent fragment. Especially we have to join especially Okazaki fragments. So this is done by DNA ligase, which is done with phosphodiesterase bond. So this is primase and DNA ligase. And this is the very important enzyme in DNA replication. This is DNA polymerase. So it will initiate DNA polymerization. So it is very important in polymerization of DNA. The deoxyribonucleic acid, or the deoxyribonucleotide, add to the 3 end of the primer, 3' end of the primer. So continue polymerization of 5' to 3' end is there. So it is continuous in the 5' to 3'. So sometimes with the complementary nucleotide polymerization, there may be errors. So we have to go in with the errors. So that place there's a proofreading activity of the DNA polymerase itself. So it has own proofreading mechanism. So what happened there? So the DNA polymerase can identify the mismatch and remove the incorrect nucleotide. That is, you call it as a exonuclease activity, and again continues the polymerase activity. So initiate of the DNA polymerization, identify the mismatch. So it's a proofreading activity, and also same as this DNA polymerase enzyme also activating DNA repair, which is similar to proofreading activity. It is happening after the replication process. So proofreading happening while in the same place, and the DNA repair after the later part of the DNA replication. And also DNA polymerase identify the DNA RNA hybrid which is at the where you have the primer. So DNA polymerase will remove the ribonucleotide as the DNA RNA hybrid and replace that with deoxyribonucleotide. So it will replace RNA primer with DNA. But there's no connection between Okazaki fragments. That is done by DNA ligase. Right. So what is the protein which is helping in DNA replication? That is single strand binding protein. As we know, DNA complementary strand. So when you are untwisting, you are not untwisting DNA strand, you will have two complementary DNA strand. So when you keep away both strands, they tend to come and get each other. So we have to prevent that thing. This thing is happened by this thing is happened with single strand binding protein. So protein molecule bind to exposed single strand DNA and it will prevent repairing and stabilize them for replication. So this is the process of DNA replication in this picture. So with the picture we can summarize the whole DNA replication process. This green color helicase is the enzyme which is separating the two DNA strand, and topoisomerase, as we discussed, it is helping us to untwist the DNA strand in the parent DNA strand. So it can cut and replace again the DNA strand. And primase will produce the primer. Primer is RNA primer is there. So to the primer, DNA polymerase will put the deoxyribonucleotide. Nucleotide, you put the nucleotide by DNA polymerase. And last part, DNA ligase will bind the Okazaki fragments by phosphodiester bonds. So, what are the similarities and differences in prokaryote and eukaryote DNA replication? So, if you take prokaryote DNA replication and eukaryote DNA replication, the process is similar, but there are differences. Prokaryote have only one origin. That means origin of replication is only one. But in eukaryotic DNA, they have several origins. The structure of the DNA polymerase is different in prokaryote and eukaryotes. Also, prokaryote replication is continuous process. Eukaryote only have the DNA replication in S phase of the cell cycle. We have discussed this one in mitosis lecture also. Right. There is the story of DNA replication. Thank you. You can come to my video in AL Biotutor. Also, you can visit my website mybiotutor.com. Thank you.